So next up, we have Melodi from MIT, and she'll be covering some of the other applications where exogenous probes can be used in respiratory disease. She's a PhD candidate in medical engineering and medical physics through Harvard MIT Health Sciences and Technology program. Her work is focused on creating diagnostic tools for infectious disease. And after finishing her studies, Melodi is hoping to work at the intersection of climate change and health. So thanks for the time today, Melodi, and we look forward to your presentation.
Hello, everyone. My name is Melodi Anahtar. I'm a senior graduate student in Sangeeta Bhattia's lab at MIT, where for the past several years, I've been developing protease-based diagnostics for pneumonia. Today, I'm going to present some of the work that I did alongside Dr. Leslie Chan, who was a postdoc in the lab, to create novel nanoparticles that can be used to diagnose respiratory infections via breath. For many years, our lab has had an interest in the early detection of cancer, as it has been shown that catching cancer in its early stages has a profound impact on survival rates. The most traditional way of screening cancer is with imaging, whereby you look for changes that might reflect cancerous growth. If there is an abnormal finding, a follow-up biopsy of the abnormal tissue is taken and analyzed by a pathologist to get a formal diagnosis. However, a more specific and efficient method of diagnosis is to look for cancer biomarkers, which are biological molecules derived from body fluids that are indicative of cancer. To this end, liquid biopsies are being developed, which involves drawing blood and looking for biomarkers like DNA, which is shed from tumors, cancer-derived metabolites, or even circulating tumor cells themselves. However, it's often said that looking for these cancer biomarkers is like looking for a needle in a haystack, as our 5 to 6 liters of blood dilutes the sought-out molecules. This is an inherent limitation of using endogenous biomarkers to diagnose disease, and it is something our lab has been trying to overcome by creating diagnostics that make their own biomarkers. To do this, our lab has leveraged human proteases, which are enzymes that are known to play an important role in processes such as inflammation, tissue remodeling, and signal transduction, and have been gaining attention as potential drug targets for infectious diseases. Our lab has designed sensors that we call activity-based nanosensors, or ABNs, to detect conditions such as thrombosis, cancer, and bacterial infection. Each ABN consists of a polyethylene glycol core that is decorated on its surface with peptide substrates. The sequences of these peptides are designed to be cleaved by specific proteases that are dysregulated in the disease microenvironment. Upon interaction with these target proteases, the peptide linkers are cleaved, which releases a small mass-encoded reporter into the blood. From there, these reporters are circulated to the kidneys, where they are filtered and concentrated in the urine. We can then take this urine and look specifically for our reporters using mass spectrometry to create signatures of differential protease activity, and therefore different disease profiles. We've been able to use these nanoparticles to not only detect cancer, but also other diseases, such as liver fibrosis and pneumonia, because of the involvement of proteases in so many different biological processes. While we have been able to accomplish a lot with our urinary nanosensors, including starting human trials, we figured we could make this technology even faster, less invasive, more accessible, and more cost-effective if we used a breath-based readout rather than a urine-based one. I don't think I need to expound on the benefits of breath, many of which are listed on this slide, to this crowd, but one limitation of current breath diagnostics as a whole is that there are thousands of endogenous volatiles that are produced naturally by the body, and the composition of each individual's volatilome is impacted by factors like the environment they live in, the drugs they take, the foods they eat, their microbiome, etc. As a result, while breath collection is certainly one of the most non-invasive sampling methods, creating useful diagnostic classifiers from breath samples suffers from poor sensitivity and specificity, which limits their clinical utility. To overcome this limitation, the breath diagnostics field has been embracing the concept of induced volatilomics, whereby exogenous volatiles are detected in the breath in order to improve the signal-to-noise ratio. Today, I'm going to discuss how we have used induced volatiles to detect acute respiratory infection in the context of bacterial pneumonia. We have modified our urinary nanoparticles to produce a breath-based readout by keeping the same scaffold and substrate scheme, but replacing the mass-encoded reporters that I had mentioned before with volatile organic compounds, or VOCs. These VOCs are linked via a peptide bond to the substrate linker and are designed to be released from the nanoparticle after cleavage by target proteases in the disease microenvironment. Once freed, the VOCs partition into the airspace of the lung and are exhaled. We then collect breath and use mass spectrometry to measure the volatile concentration. Using this method, we can create a breath signature of disease that is distinct from the signature generated by healthy controls. We term these particles volatile ABNs, or VABNs, as I will be referring to them from now on. To create our proof of concept, we designed our nanoparticles to be cleaved by neutrophil elastase in the context of bacterial pneumonia. We had previously shown that neutrophil elastase activity was elevated in this model using our urinary ABNs. Furthermore, it was important to us that the substrate we used for these pilot studies was cleaved at the C terminus, so that the reporter could be released from the nanoparticle without any additional amino acids attached, which would presumably affect its volatile properties. A group had previously published a fluorescent substrate for neutrophil elastase that was cleaved at the desired site, making it a natural starting point for our work. As for the volatile reporter, in order for us to attach the reporter to the peptide substrate, we needed a VOC with a terminal amine group that could form a peptide bond. We also needed to ensure that the VOC was not produced naturally by the body and had low environmental levels in order to optimize our signal to noise. Based on these properties, we eventually selected hydrofluoroamines as our reporter molecules. The structure of the probe is shown here, with the neutrophil elastase substrate in black, the attached hydrofluoroamine in gray, and its protease-liberated form in orange. To determine whether this chemistry would enable protease-mediated VOC cleavage, we first added pure neutrophil elastase and VABNs with our substrate VOC pair into a 20 ml vial. Our thought was that over time, neutrophil elastase would cleave the substrate, which would allow the VOC to accumulate in the headspace of the vial. We then sampled the headspace and observed that in the absence of neutrophil elastase, we did not see any signal corresponding to the volatile. But when we added neutrophil elastase, we immediately saw a spike in signal corresponding to the VOC. This was our first indication that our substrate could enable us to detect neutrophil elastase activity via a volatile readout. We also analyzed the liquid fraction of the vial containing the diluted enzyme in probe and validated that in the presence of neutrophil elastase, the probe was cleaved and the VOC was able to partition out of the liquid fraction and into the gaseous headspace. While these experiments had shown that our nanoparticles could work in the perfect world where it was directly in contact with its target protease, the VABN's performance in vivo was still unknown. To try to predict how the complex in vivo environment would affect exhaled reporter levels, we created a physiologically based pharmacokinetic model. This model consists of three main compartments: the respiratory lumen, the respiratory tissue, and the breath collection chamber. Because we administer the VABNs intratracheally, we assume that the full dose of the VABNs would be present in the respiratory lumen at time 0. Over time, the VABNs would then diffuse into the tissue, where they would either be cleaved by proteases or cleared into the blood. After being cleaved by proteases, the nanoparticles would release their VOCs into the tissue. From there, the VOCs could diffuse into the lumen, after which they could be exhaled or be cleared into the blood. By inputting VOC properties such as the partition coefficients, which describe how readily the VOC would partition into blood and tissue versus air, the Michaelis-Menten kinetics of the VABN, which is substrate and VOC dependent and can be measured in vitro, and by fitting to breath measurements from pilot in vivo experiments, we could predict how changes in the system would affect breath signal. For example, we could model how changing the formulation of our nanoparticles from a nanoparticle to microparticle to free peptide format would impact breath signal. We could predict what dose we would need to deliver in order to achieve detectable breath signal. And we could even estimate how our ABNs would perform in humans by using allometric scaling. These analyses allowed us to settle on a final formulation and dose that we were able to bring forward into our mouse experiments. Now that we knew that our VABNs could produce a detectable signal, we needed to perform in vivo experiments to evaluate their diagnostic potential. The experiments I'll describe in the next few slides all use the same framework, whereby we delivered VABNs into either healthy control mice or mice infected with Pseudomonas aeruginosa, which is our model of bacterial pneumonia, In this model of bacterial pneumonia, there is increased neutrophil elastase expression compared to healthy mice. We then collected breath at various time points and analyzed the samples using mass spec. The resulting peaks look like the traces here, where you can see that there are large peaks from mice that were infected with pneumonia and much smaller peaks from the healthy controls. In many cases, it was quite clear even before quantification which mice had a significant reporter signal and which did not. By creating standard curves that correlated the mass spec measurements to absolute concentrations, we were able to quantify the levels of our volatile reporters in exhaled breath. Then, by collecting breath longitudinally, we were able to see that there is a clear washout effect, whereby we saw peak signal around 10 minutes after ABN administration that continued to decrease over the next 3 hours. We also observed that the concentration of reporter in the breath was dose dependent. Furthermore, there is a clear difference in breath signal between the healthy and infected mice, indicating that the signal to noise ratio is quite high. Protease activity is dynamic and can change over the course of infection. So in order to see whether our VABNs could not only detect changes in protease activity over time, but also monitor changes in disease, we performed an experiment where we infected mice with Pseudomonas and subsequently treated them with ciprofloxacin, an antibiotic. To do this experiment, we first took baseline breath measurements from all mice at day 0. We then infected a cohort of mice with Pseudomonas, abbreviated here with PA01, and represented with the red triangle. 8 hours after initiating the infection, we administered our first dose of VABNs and collected breath, a process represented by the white triangle. 16 hours later, we collected breath again, and then treated mice with a 2-day regimen of ciprofloxacin, represented by the blue triangles. After giving the mice time to recover, we then collected breath again at days 4 and 7 to see if the ABNs could be used to monitor treatment. When we looked at the breath concentrations, we saw that at day 0, all mice had approximately the same level of signal. This represents the level of noise that we would expect with our VABNs. Then, 8 hours after an infection was initiated, mice that were infected with Pseudomonas had a significantly increased breath signal. This high level of breath signal was maintained 24 hours after initiation of infection, at which point the first dose of antibiotics was given. At day 4, after mice had received both doses of antibiotics, the breath signal in the infected mice had returned back to baseline levels, indicating that the VABNs were detecting changes in protease activity that reflected disease over time. We can evaluate the ability of our VABNs to distinguish disease from healthy controls by plotting the results with receiver operating characteristic curve, as shown here. A test with perfect sensitivity and specificity would have an area under the curve, or AUC, of 1, whereas a test that has classification that's as good as chance would have an AUC of 0.5, which is shown here with the red dotted line. As you can see, at the first 2 time points where the infection was raging in these mice, the VABNs were able to perfectly distinguish between mice with infection and those without. Even 4 days after infection, we were able to detect differences between these groups. It's only by 1 week after infection that the classification power really wanes. We then wanted to demonstrate that our VABNs could do more than just detect infection. To do this, we focused on alpha-1 antitrypsin deficiency, or AATD. Normally, alpha-1 antitrypsin, or A1AT, helps maintain pulmonary integrity by inhibiting proteases that are produced by neutrophils in response to insults, such as smoking, inflammation, etc. In AATD, this inhibitor is not present, which means that the neutrophil-derived protease activity, like that of neutrophil elastase, is unchecked, causing damage that can result in conditions such as emphysema. To treat AATD, people are regularly administered alpha antitrypsin, which helps their bodies maintain proteolytic homeostasis. We sought to demonstrate that our VABNs could be used to monitor how effectively A1AT was controlling inflammation by detecting aberrant neutrophil elastase activity. To do this, we administered lipopolysaccharide, or LPS, into wild-type mice and those lacking A1AT. LPS is a bacterial-derived component that is used to initiate inflammation without the use of pathogens, creating what's known as sterile inflammation. Whereas wild-type mice would be able to control this inflammation by inhibiting neutrophil elastase via A1AT, the antitrypsin knockout mice would not be able to inhibit activity, leading to lung injury. To determine whether our VABNs could detect this difference in neutrophil elastase activity, alpha antitrypsin deficient mice were given VABNs 3 days before initiating inflammation with LPS to get a baseline reading of breath signal. The day before giving LPS, some of the mice were treated with A1AT, as shown by the blue triangle. Then 1 day after the LPS challenge, the ABNs were redosed into the mice to see if the breath signal in the treated mice was different than those without antitrypsin treatment. The baseline levels at day -3 were equal across the board for all wild-type mice, represented with circle symbols, and A1AT deficient mice, which are represented with squares. At day 1, wild-type mice that were challenged with LPS, the red circles shown here, had significantly increased breath signal corresponding to increased neutrophil elastase activity as measured by the VABNs. A similar trend was also observed for the A1AT deficient mice, shown with the squares. Interestingly, the LPS-treated knockout mice had significantly higher breath signal compared to the treated wild-type mice, indicating increased neutrophil elastase activity. Importantly, treating the A1AT deficient mice the day before LPS challenge lowered their breath signal to the same level as the treated wild-type controls, which is indicative of lowered neutrophil elastase activity, presumably due to inhibition by A1AT. Looking again at the ROC curve, we saw that the breath signal was able to distinguish between inflammation in wild-type and knockout mice with high sensitivity and specificity, once again highlighting the impressive signal-to-noise ratio capabilities of the VABN. Having shown that the VABNs are capable of generating a rapid readout of protease activity that is sensitive and specific, we are now looking forward to new diseases that could benefit from rapid non-invasive detection. Naturally, given our interest in respiratory disease, our thoughts turn to lung cancer. It has been well established that detecting lung cancer in its early stages can dramatically improve survival rates. Acting on this finding, people at high risk for lung cancer have begun being screened regularly in order to catch cancer early and stop its progression in its tracks. This screening is done using low-dose CT, which suffers from two main problems. The first is that false positives are a frequent issue due to the high sensitivity of CT. The second is that each scan exposes the patient to radiation, so screenings must be somewhat restricted in order to limit radiation exposure. Ideally, everyone would be able to get regularly screened for lung cancer, regardless of their risk status. But in order to do this, the world needs a screening method that is highly sensitive in order to pick up the cancer in its early stages, highly specific, such that the test can tell the difference between cancer and other respiratory conditions such as infection or COPD, and non-invasive to enable safe and frequent testing. Previously, our lab used our urinary ABN to detect lung cancer in its early stages, in many cases matching or exceeding the capabilities of CT imaging. Now we are working on adapting our volatile ABNs to create an analogous system that can be used to detect lung cancer via breath. If successful, this test would check off all the boxes for an ideal lung cancer screening tool. We hope that those who are interested in joining our efforts will reach out throughout the conference, and we would love to hear from you all. I'd like to end my talk by thanking all the great scientists in our lab and our collaborators who contributed to this work. Namely, I'd like to thank Dr. Leslie Chan, who spearheaded the animal studies that I presented, as well as our funding sources. Thank you all for your time, and I'm happy to try to answer any questions you might have in the following Q&A session or offline.
We've got time for a few questions. First for me is more of an observation. I remember being really excited reading your papers in the sense that, by being able to choose the volatile reporter, it being a unique compound obviously means that you don't need to worry about background quite as much. You can have a higher concentration. But I think one of the other things that struck me was that you can then pick and choose the volatile reporter to then be more easily detected by different types of technology. I know within our mobility, one of the problems that we see in the breath field is a lot of the endogenous compounds, tend not to be ionized by our mobility devices relative to background. But with some of the volatile reporters you picked there, they would be very easily ionized relative to complex background. So I do think there's so many advantages of the approach that you've taken there. So the first question is, how widely do you think this approach could be applied? Is it relevant for all illnesses? What are the types of limitations?
Yeah, so I kind of answered this already by responding, but I can expound on it a bit. We think that the power of the ABNs is really that because we can change the peptide sequences to target, any protease really, that this can be applied to a wide range of diseases. So for this particular model, we knew that neutrophil elastase was implicated and there was a really good substrate for it. So we were able to target it that way. Other possibilities are that, microbes themselves can produce their own proteases, and there are, it's possible that we can change that peptide substrate to detect specific bugs rather than the host response. But then in addition, the host response itself, there are over 500 human proteases, so we can target each of them as long as, that sequence that is specific and sensitive enough is available. So that's the real limitation, is that you need to have the right substrate. But once you do, we think that this can be applied to a ton of different diseases.
Brilliant, thank you. Next question from Christian. Wonderful presentation. Do you think the addition of volatile reporters would limit the amount of proteases that could be targeted? For instance, only proteases that cleave the terminal sites would release a reporter without affecting its volatility.
Right. So we're actually looking into this more. So the way that the form of the probe that I presented in this work, it's really dependent on that terminal cleavage, but we're working on creating different types of chemistries that can enable cleavage, somewhere within the substrate, not necessarily at the end, and then allow for the unmodified release of the volatiles after. So that's chemistry that we're currently working out, and that would allow us to expand our pool of volatiles and also our pool of substrates.
Brilliant. Next question is from Scott. Is the use of fluorocarbons deliberate to help availability to the enzyme?
The idea of using fluorinated compounds was just because it's inert and not natural. I don't think it was deliberate for the use of neutrophil elastase in particular, but if that ends up being the case, it would be a happy kind of coincidence. So that's also one of the strengths of being able to modify the volatile. If there is some kind of inherent advantage to using one over the other, then that's something that we'd be able to incorporate into these probes.
Got it. So it's more just about the uniqueness of that compound relative to other things you might find in the background. That's great, thanks. Next question here. I think it's been posted. Sorry, it's been posted in the chat, not the Q&A. Will this technique be able to differentiate bacterial infection and chronic lungs, such as COPD, where lung protease activity is high?
So I think this actually ties into one of the other questions in terms of multiplexing. So, I mean, with one probe, it's difficult to get that kind of specificity because you're really just relying on the expression of one protease. But we imagine that our VABNs would be able to be multiplexed the same way that we do with our urinary versions, where we can administer 20 of them at one time. And then by looking at the readout, we can create different signatures of disease. And because those specific profiles of proteases are pretty sensitive and specific, we're able to stratify different diseases based on the different sets of reporters that come out. So we haven't enabled that technology for the breath work yet. We're working on multiplexing, but once, we imagine that if it can work the same way as our urinary reporters, we would be able to differentiate between different lung conditions and then just different diseases in general. Obviously, the imaginary dream world is coming up with a panel of proteases that can distinguish between all types of different diseases. But we're taking it one step at a time. And right now, lung cancer is our next target.
Got it. Thank you. I think the question popped up in the previous session around some of the regulatory implications of probe administration. So I was wondering if you might be able to comment on that, particularly if you're going down a multiplexing type route, for instance, what the kind of regulatory implications of that are.
Yeah, so one of the advantages of this system is that for at least the urine form, that's already kind of gone through approval because it's been tested in humans already. So that in terms of the regulation, that's already been approved. It's more of a matter of the volatiles being safe for people. So there are different classes of volatiles, obviously, that are more harmful than others, and it's just a matter of choosing ones that are safe. So going forward, we're thinking of ones that are, part of food additives or fragrances or something that's already kind of approved. And we figure if that volatile is already known to be safe for humans, the regulatory path is pretty straightforward because we already have existing technology that's gotten approved before.
Got it. Thank you so much, Melody. That was really wonderful, inspirational work. So thanks for joining and taking the time to present and answer questions today.